The aim of this experiment was to analyse the protein content of wheat and barley seeds.
There are approximately 20 wheat and barley seeds laid out on trays with moisture to trigger germination, and the germination halted at different time periods, and these time periods include 6 hours, 12 hours, 24 hours, 36 hours, 48 hours and 60 hours.
To halt the germination of the seeds, we placed the seeds in trays and put them in the incubator for 20 minutes at 80 degrees Celsius. When the time was up, we took them out of the incubator.
For an after-school excursion, we went to Murdoch University to start analysing the wheat, and what we did was we crushed the seeds to extract the protein, and we then used a centrifuge and a solvent to break down the particles, and we pipetted it onto a metal plate and got it ready for the mass spectrometer.
The mass spectrometer is a piece of technology that energises proteins on a plate. This causes the proteins to fly or travel up a tube to hit another plate. The mass spectrometer measures the time taken between these two plates, and this is then able to provide us with information about the mass of protein molecules.
Once we had the data on the protein content on the seeds we germinated for different amounts of time, a protein database was used to correlate between the protein in the seeds and their allergenic properties.
Once the mass spectrometer had run, it provided us with a vertical bar graph called a mass spectrum, and this graph highlighted different peaks of proteins, the largest being the protein called the base peak with an abundance of 100% at that point of time. And from this, we were able to determine the allergenic symptoms of those proteins.
The main protein we identified was hortine, although it has many variations. So when hortine is mixed with gliadin, it forms the general name gluten. And for celiacs and gluten intolerance, they have many severe symptoms.
So during the germination process, proteins in the seed are used up and digested. So this means that there are less allergenic properties in the seed after germination. For future use of this technology, it means that bakers will be able to use this process to ensure that the doughs they use don't contain as many allergenic properties, so people that are intolerant to gluten can digest it safely.
Finally, sourdough bread has less allergenic properties for similar reasons.
And this is because the dough is fermented, which means that the proteins are used up and there are no longer allergenic properties in it.
So during the process, the mass spectrometer actually broke down at Murdoch University, and so we had Professor Florides, who was our mentor during this, have to tell us that we couldn't continue the research, and from this we were able to distinguish and really identify with the cost of this machine, and when it breaks down, PhD students, master students and all these agricultural companies couldn't continue their research. So it's a good experience to learn about what happens when this does, but I overall really enjoyed the experience, and it gave me the opportunity to do something that I don't get to do during class time.
Personally, I really enjoyed the whole germination process.
And preparing the seeds for analysis, and I think that I also really enjoyed using the equipment that they had there at the university and seeing what research projects are like at that level.